Gabriela Timoney, and like Dr. Hoke said, I work in Dr. Boland's lab. And so today I'm going to talk about my research, which is regarding the structural basis for the effects of fatty acids on the function of voltage-gated potassium channels, otherwise known as Kv4 channels. So what are Kv4 channels, and why do we care what they do? Why do we bother to study them? So Kv4 channels are located in postsynaptic regions of the dendrites. And this means that they have the potential to control synaptic outputs. Essentially what they do is they modulate the summation of excitatory postsynaptic potentials, EPSPs, and modulate synaptic outputs. So you might be asking, what does that mean? Essentially, these channels control how frequently neurons talk to each other, how they pass information. So as you can see from this figure here, all right, that's when Kv4 channels are functioning. They really modulate how these neurons fire. So right here, it's twice in this period of time. However, if you block these Kv4 channels, say with the toxin from the huntsman spider, there's increased neuronal firing. So essentially, when these Kv4 channels are blocked, EPSPs are allowed to accumulate, summate, and increase the firing rate between these neurons. So why is that important? Well, each time a neuron fires, information is transferred. So if neurons are fired more frequently, more information can be transferred, and this is really important in the processes of learning and memory formation. So that's why we study these. But instead of using a toxin, we use fatty acids. And fatty acids are naturally found in the brain. They're released by high-frequency stimulation of glutamatergic receptors in the brain. And these polyunsaturated fatty acids, PUFAs, are homolytically cleaved inside the plasma membrane. So you have these fatty acids in your plasma membranes, and what they do is they can block these Kv4 channels. If you look here, this green trace is a wild-type trace. However, this red trace right here, as you can see, there's inhibition right there. These fatty acids block those Kv4 channels. They block that peak max effect right here. And you might ask, what is this? This is a trace. And so right here, these channels are opening. They open transiently and they shut very rapidly. Some channels don't shut, some stay open. The ones we look at close very rapidly. And essentially these fatty acids inhibit how much current is passed between the internal and external cellular regions. So how do polyunsaturated fatty acids block these channels? How do they inhibit these channels? That's what we're trying to figure out. We know that it happens, but we don't know the mechanism by which it happens. So we use an oocyte expression system, which is essentially we take oocytes from xenopus frogs, we do surgeries on them, and we inject them with RNA. And inside these cells, the RNA is turned into proteins, and it's folded and everything right inside the cell. It's nice little, it's like a protein factory. And they're transported into the cellular membrane, into the plasma membrane, where we can then test them using 2-voltage clamp electrophysiology. And what we do is we put 2 electrodes in the cell, and one electrode measures the current, and the other one injects current into the cell. And we measure how the cell responds. So what RNA do we inject? We mutate RNA, as you'll see in a second, and that's how we kind of test. We make these mutations to see, well, what happens if we change this amino acid? How does this change how the protein folds? How does this change how the fatty acids interact with these channels? Okay. So, as I mentioned, we do mutagenesis reactions. So we make point mutations, which means that we change one base pair of an amino acid. Amino acids, if you remember, are encoded by 3 like nucleotides. So if you change one, you can change the amino acid. So to start, we were reading papers, and there's a study done in an IK1 channel, which is a similar but unrelated channel because it's not regulated by voltage. And in this paper, they made a mutation in the pore region of the channel. Essentially the region of the channel where ions pass through. So you have this protein sitting in the membrane, but only certain parts of that are actually exposed to the channel. To the pore region. So what they did was they made a mutation they mutated one of the amino acids in that pore region, and consequently there was a lack of inhibition by fatty acids. So what we decided to do was make a similar mutation in this Kv4-2 region, and it's really interesting because in this region it's conserved throughout all potassium channels. So voltage-gated, inactivating channels, inward rectifiers, outward rectifiers, all these potassium channels all have this same sequence of 8 amino acids. So if this is conserved, if this is passed on from from species to species or like type to type, then it must be important. So as you can see here, here's our Kv4-2, and the gray regions show areas of similarity between different channels. And this is the mutation we made right here, our first mutation, the T370. We call it T370 because it is the threonine at the 370 location, so the 370th amino acid. So if you look here, this is a crystallized structure of KCSA, which is a type of channel. Channels, in order for you to get a good idea of what they look like, have to be crystallized. Kv4-2 hasn't been crystallized yet, but this is really similar to Kv4-2, so we can use it as a model when we're trying to figure out where we should mutate. So our mutation is this orange mutation right here. This is the extracellular region of the cell. This is the transmembrane domains. So this is sitting inside that plasma membrane, and then this is the intercellular region. And then this is from the outside, and this was crystallized in the closed state. You can't really choose what state the channel's in when you crystallize it. You just crystallize it and then you realize what state it's in. So this is crystallized in the closed state, so that's the view from the outside, and this is the view from the inside of the cell. And what's really interesting is you can see that in that view from the internal side down here, you can see that threonine location that's exposed. And in a paper we just wrote, we show that we believe, and we have strong evidence to prove, that fatty acids actually do work from the internal side of the membrane. They get into the cell and then interact with the pore region, as opposed to interacting from the outside. Okay, so as I showed you earlier, here's that control trace, and here's when you apply fatty acids. So you see about a 60% inhibition of peak current, typically, or 60% inhibition of peak current. However, in the mutant T370, there's a significant lack of fatty acid inhibition. So essentially, you're only seeing about 15 to 20% inhibition by fatty acids as opposed to 60%. And this is huge. You might notice a little bit too here. There's like a kinetic effect that I won't go into today. But this mutation also changes the rate at which the channel's closing, which is important later when we talk about our other mutations we're making. But as you can see here, this mutation does have a significant effect on the channel's ability to interact with fatty acids. So another consequence of our mutation, another result that we saw from this mutation, was that here's your wild type, and it starts to inactivate at about 20 millivolts, which is 20 millivolts. So what we noticed was that in the T370S mutation, there's a leftward shift in activation. What does that mean? That means that the cell becomes more sensitive to voltage and starts to activate at more hyperpolarized levels. So when your wild type is at 20 millivolts, you have almost complete activation of the mutant channel up here. So that was really interesting because that was a very unexpected result, and we weren't sure why that was happening. And it was really disconcerting. We thought maybe we had done something wrong. But we went back through the literature, and we found that in a paper written about 10 years ago, by a graduate student, Rob McKinnon's lab. I don't know if you know Rob McKinnon. He won the Nobel Prize in 2003. So he is like the godfather of potassium channels. A paper from his lab made a similar mutation when they were testing the selectivity of these channels. Now I said this is a voltage-gated potassium channel, which means it's very selective to just potassium channels that flow in and out of the channel. So they made these mutations to see how that affected which ions could pass through the channel. Could sodium ions pass through the channel or chloride ions? So that's what they were testing. But they made this similar mutation where they changed a threonine to the serine at the same position, and they too see this left shift right here in activation. And they don't explain it. It's simply like in a small figure on the last page of the paper, but it's there. So that was reassuring to us because even if we don't know why it's happening, we know that someone else has seen the same results. So, yeah. So where do we go from here? We've made this T370S mutation, but we need to know more about this mutation. We need to make more mutations to figure out what's going on. So we're making this T293A mutation, and this mutation is in the voltage sensing region of the pore, also known as the S4 region. And this region is what conveys that voltage sensitivity to the channel. So when you make a mutation here and change this threonine, which is a polar molecule, to an alanine, which is a nonpolar molecule, you create this left shift in activation, and the channel again becomes sensitive to voltage. So what we want to see is if we make this mutation and left shift this activation in a channel that we know will left shift from this mutation, will it still be inhibited by fatty acids, or is the reason that the T370S mutation, the reason that there's a significant lack of block by fatty acids, is that because there's this left shift in activation? Is that somehow creating this effect that we see? So our preliminary results show that T293A is in fact sensitive to polyunsaturated fatty acids, which does suggest that this left shift in activation of T370 is not the reason for the lack of fatty acid inhibition. These three mutations, T370V, T370L, and T370A, are all being made because we want to make sure that the results we're seeing aren't a result of mutating that serine to a serine. We want to make sure it's because there's no serine there that we're seeing our results, and not because there's a serine there. These other mutations are similar. They're kind of checks for us. As I talked about earlier, we saw that change in inactivation, change in the rate of inactivation. So this delta 2 to 40 T370S is essentially lacking a part of the channel that is kind of like a tail. You can attach things to that tail, and they change how the channel the rate at which the channel expresses and how it inactivates. So we deleted that section of the channel, and that results in a slower inactivation. And the T370S mutation has a slower activation than the wild type. So we wanted to see, well, is this slower activation somehow resulting, again, in this lack of inhibition by fatty acids? So that's why we're making that mutation. This B395A mutation was also made in the same paper that we found the T370S mutation in. And they showed that it conferred some insensitivity to fatty acids. So when they made this mutation, there was a lack of inhibition by fatty acids on the channel, but it was not as significant as the T370S mutation. And this is important because this isn't located in the pore region. All these mutations we're talking about are located in that pore region of the channel, and this is located outside. So it's interesting for us. We want to test it. We want to see, well, if we're concentrating on the pore region, then why is this other site important too? The final mutation, T369S, is being made because if you remember back to that chart I showed you of that 8 amino acid sequence, it's TTVGYG, and we're mutating that second threonine. But there's also a threonine at the 369 spot. So we want to make sure that that threonine isn't important in conveying sensitivity. We want to make sure that we've pinpointed that location. That specific threonine is what's important. So again, why is this important? And why do we care about this? These channels are really important in regulating neuronal firing, like I said. So by figuring out the mechanism by which your body controls how often a neuron fires is really important in those processes for learning and memory. And they have pharmacological implications with Alzheimer's and learning disorders. So that's why we want to figure out the physiology behind that.